I didn't look.
Start because it's time. So, good morning, everyone. Today we have the pleasure to present a paper called "Generation of Functional Oligopeptides that Promote Osteogenesis Based on Unsupervised Deep Learning of Protein EDRs." It is a kind of mixed journal club, computer science and biotechnology journal club. And Hannah Ford will present this journal club. She is a placement student in bone biopathology lab here in the University of L'Aquila, handled by Professor Hannah Teti. And she is here more than 6 months now. So yes, Hannah will present his journal club, and also Professor De Gasperis will help us with the discussion because he's an expert on artificial intelligence and computer science. So he can explain better than us how it's work. So yeah, that's all. I will leave the floor to Hannah and see you in the discussion.
Thank you, Michaela. I'll just share my screen. So, can you see? Right. So yes, the paper that I'll present today is called "The Generation of Functional Oligopeptides that Promote Osteogenesis Based on an Unsupervised Deep Learning of Protein IDRs." So first, some basic background on protein structure. Proteins are made of amino acids, which have different components, one of which is a variable side chain, which can have different properties, such as being hydrophilic, meaning that they dissolve well in water, or hydrophobic, meaning they don't. Hydrophobic amino acids tend to reside on the inside of the protein and give the protein its rigid structure, whereas hydrophilic amino acids are able to not do this. The primary structure of a protein is the amino acids bonded together by peptide bonds, and then this folds to produce a more complex structure of proteins. Now, IDRs, which are the regions of proteins which this paper focuses on, are characterized by not assuming one structured shape. So they are able to be found in many different organizations, kind of. They have a low sequence complexity of amino acids, and they are enriched in hydrophilic amino acids. So these ones are those that can dissolve well in water. They tend to be around 3 to 10 amino acids long, so they're quite short. And this paper chose, the researchers who made this paper chose to study these because of the increase in recent data in the literature and emphasizing how functionally important these regions are. They are involved in many functions, involving protein-protein and protein-nucleic interaction, as well as other things. I've put one example here of a super enhancer because this is a region where the IDRs of transcription factors interact with each other in order to regulate mass gene expression, for example. But there is still a lot that remains unclear about the specific roles and how these regions of proteins specifically function. Some more background on the bone. So the bone is composed of many different cell types. The hematopoietic stem cells develop into macrophages and osteoclasts, which are the bone resorbing cells. And the osteoprogenitor cells, the mesenchymal stem cells, can mature into pre-osteoblasts and osteoblasts. And the most mature and differentiated form of this cell is the osteocyte, which is the cells that make up the majority of the bone. This process of maturation and differentiation can be defined by the expression of different markers, such as SOX9, RUNX2, ALP. And so as you go along the process of differentiation and maturity, there are different markers for this. So for example, an ALP positive cell is more mature than a non-ALP positive cell. So in this paper, they aim to design a small peptide, less than 10 amino acids long, based on the IDRs of osteogenic proteins, so proteins that have known function in bone formation, using an unsupervised deep learning model, which is the bioinformatics section of this, which can promote bone formation. So the two parts of the study, firstly is the informatics section. So how they did this is they took the composition of amino acids found in the IDRs of the osteogenic proteins. They ordered the amino acids in terms of their frequency in these regions, and then they generated new oligopeptides based on this data, and they used software such as Engrams and Monte Carlo to extend the oligopeptides. The oligopeptides that were produced and the ones that had the higher probability vectors were then selected for further experiments. 28 oligopeptides were then tested in vitro, with the most effective inducers of bone formation being used for further experiments. And they selected one final peptide, which they named AIB5P, which had the strongest osteogenic activity, and they used this for the next in vitro and in vivo analyses. So first, I will give my best attempt at explaining the deep learning technique, because this is something I was not familiar with before. But it's a type of artificial intelligence, and it mimics the way that humans learn. So you give it a data set and you say, these are the conditions that we want to reproduce. And then it learns the techniques and how you reproduce this. And then when you give it new data, it is able to use its knowledge to produce things that relate to what it has learned before. So the neurons are indicated by these circles, and these are just things that hold a value. And each layer is a new layer of processing. Each neuron makes a decision based on the results of the previous neuron of the previous layer. And yes, the system can then select new data based on what it has already learned. This can be explained in a way of using the example of handwritten letters. Now, you can see that these are all the letter 3 in a very pixelated way, but a computer is based off numerical data. So this would be the information that the computer would receive. So it's a letter 3 by the values and the spread of the positive signals from each place that the 3 is covered. And it is able to know that this is a 3, and also this is a 3 and this is a 3. So you are, by showing a computer many examples of something that is one value, it will learn to recognize different versions of this, even if the values are very different. This is this thing kind of visualized. So this 784 refers to how many squares there are in this square where the 2 is written. And then through processing through the layers, the machine can then come to the conclusion that this is in fact a 2 and not a 4 or a 5 or a 6, which seems simple to us because you can see that this is a 2. But for an artificial intelligence to be able to do this, it's quite impressive. So in this case, we're not talking about numbers, we're talking about proteins. So they took a protein data set and found proteins that were implicated in the terms ossification, osteogenesis, osteoblast development, and osteoblast differentiation. From this, they collected 171 osteogenic proteins, and they used this for their data set. They used the software IUP-RED2A to identify the IDRs of these osteogenic proteins, so the amino acid sequences of these IDRs. And this was what was used as the deep learning training phase. So they used the amino acid sequences of the IDRs of the osteogenic proteins to train the machine to say, this is the kind of thing that we want. So the most upregulated amino acids were ordered and were used as the building blocks to generate the new oligopeptides. They used the Engram software, which is used to infer the probability of the residues of the oligopeptides awaiting extension. This is kind of a software that is able to predict the most likely outcome of a situation. It's used in translation devices and in map devices and even Alexa devices. And it basically says, if you give it the data of the first amino acid of the new oligopeptides, it will kind of be able to calculate what is the most likely next amino acid to come after this one. So yes, the amino acids, as you can use single letters, abbreviations for them, they were input as letters into the engrams and given a probability score based on the order and frequency found in the IDRs of the previous osteogenic proteins, I believe. So these amino acids were extended using Monte Carlo simulation, which extends them according to some more inferred probabilities. And this led to the generation of the artificial intelligence oligopeptides. So this is the process that it followed. The initial amino acid, the probability vector was considered and the next amino acid was added. And then this continued until the end condition was reached, which generated artificial intelligence oligopeptides. These would then pass through the deep learning modules to then give the outcome of the most probable osteogenic oligopeptides. These clustering trees show the output of the screening. So the bigger letters are the more probable osteogenic proteins from the output of the artificial intelligence. So these oligopeptides would have a higher probability of having osteogenic properties based on the ideas of the original osteogenic proteins that the system learned. They were grouped in either being mainly composed of serine and glutamine or glycine, proline, and alanine. And the highest probability rating oligopeptides were selected. So it's these ones written here with the highest score of frequency. So they then produced these peptides with some company and were then tested them on bone marrow mesenchymal stem cells. So the alizarin in red showing the osteogenic potency of all of these different peptides led them to select the ESSES peptide for further experiments, which they named Artificial Intelligence Developed Bone Forming Pentapeptide, AIB5P. They then used this peptide and applied it to bone marrow mesenchymal stem cells of mouse and human. And they found an upregulation in osteoblast differentiation markers. Here they used BMP2 as positive control, as this is known to induce osteoblast differentiation. And so they have, from this, they could conclude that AIB5P potentially works by inducing differentiation of the mesenchymal stem cells. This is the results of the ALP staining and the alizarin red staining, showing the osteoblast differentiation indicated by a darker blue color of both mouse and human stem cells, and the darker red color of the alizarin red staining showing osteogenic potency. And you can see that compared to the control, there was a deeper red in both human and mouse stem cells. Here was just to show that they found that the protein affected to a greater extent stem cells rather than osteoblasts. So they could achieve the deeper red and blue colors when they applied the protein to stem cells, you can see here, rather than osteoblasts. So they think this protein works most effectively by enhancing the development of stem cells. So next they did some in vivo experiments and they used LEPR positive cells, which are a major source of stem cells which contribute to bone formation in adult bone marrow. They administered AIB5P every 3 days for 1.5 months, and you can see the greater distance here is an increase in cortical bone formation. There's also a greater signal from the red fluorescence here, which is due to the newly formed osteocytes from the LEPR positive mesenchymal stem cells after the administration of the peptide that they produced. There was also an upregulation in many osteoblast differentiation markers, showing again similar results as they experienced in vitro. This here shows again, bone deposition. So the bigger gap between the green and the red line shows an increase in bone deposition. This was after 2 weeks of growth. In the control, there was a smaller distance, whereas when the protein was present, there was more bone deposition. And this is shown graphically by the mineral apposition rate and the bone formation rate, significantly increased when the protein is present. Calcium deposit levels were measured by von Koster staining. And again, you can see an increase in deposit of calcium levels, which is important for the generation of bone. DMP1 was increased after the administration of AIP5P, which is important because it's a protein which has roles in mineralization of bone. So we can see that the bone mineralization levels are increased. Next, they performed some micro-CT analysis, and they found that the peptide increases bone mass. You can see with your eyes here that there is a lot more bone when the peptide was present rather than the control. And graphically, again, the overall conclusion is that there is an increase in bone mass. They wanted to confirm that the increase in bone mass was not due to a downregulation of osteoclast activity, and which they did here. So they did some trap staining and found that when they administer the peptide, there is an increase in osteoclast activity. So the bone could be inferred to be of good quality, as osteoclasts are still resorbing bone while osteoblasts are secreting more bone. So next, they wanted to study the effect of this peptide in bone loss and defect models. So they used an osteoporosis model, a fracture model, and a titanium implant model. So first, the osteoporosis model, they used overexerized mice, meaning that they removed the ovaries of these mice, which induces osteoporosis. So they found that the peptide increased bone in the overexerized mice when you compare it to the mice without the peptide being administered. You can see it graphically here. Bone mineral density, bone volume on tissue volume, all of these parameters were increased after administration of the peptide. And here, parathyroid hormone was used as a positive control, as this also is known to increase the bone volume and bone mass. Similar results were seen from the vertebrae bone. So again, administration of the protein increases bone mass compared to the osteoporosis model. Next, the fracture model. They performed a fracture in a bone, and the protein, the peptide, was able to heal this fracture to a much greater extent than the control. You can see here the fracture has been filled in with new bone, and graphically here, the bone volume was increased. Titanium implant models, so they implanted some titanium, and again, with the administration of the peptide, you can see that the bone has healed to a much greater extent around the implant compared to the control. And here, the two stainings show this red here in the control is the bone, and the red here is the bone after the peptide. So there is a much greater proportion of bone present. So next, now they've confirmed that this peptide works, it increases bone volume in kind of a steady state and in a bone defect model. So they wanted to know how it does this. So they performed immunofluorescence targeting this peptide after it has been treated on bone marrow mesenchymal stem cells. And they found that it localizes to the cell surface. So you can see this is the fluorescence of the peptide that they received, and then this was the contrast phase. When they merged it, they could see that the peptide was residing at the cell surface. So from this, they inferred that this is having its effect by interacting with a cell surface receptor. So they wanted to know which cell surface receptor this was. So they allowed the peptide to bind to the cells. This cross-linked as it did by itself, and then they performed a protein extraction. So they could separate all the components, and then biotin was conjugated to the peptide here so that they could use streptavidin beads, which bind biotin, to separate out the peptide and whatever it is that it is bound to. Then they performed SDS-Page and mass spectrometry assay to see what is present in this collection of proteins. They found that the most commonly expressed protein in this pull down was the integrin alpha-5 protein. Here they confirmed that when the artificial intelligence peptide was present with the biotin, also integrin alpha-5 was present. This was not the same for other similar proteins with similar effects. So next they wanted to see what happens when they silence integrin alpha-5. Here, confirming the silencing of integrin alpha-5, they found that when the peptide was present and integrin alpha-5 was silenced, the effects that it has when integrin alpha-5 is not silenced were reduced. And the gene expression of the osteoblast differentiation markers was reduced to a normal level, to when the peptide was not there. So they could infer that the peptide that they produced interacts with integrin alpha-5 to bring about its effects. Again, here you can see this is kind of the positive result of the peptide and integrin alpha-5 unaffected, compared to this on the bottom, the peptide here, integrin alpha-5 silenced. The results are comparable to the control with no peptide present. Next, they looked at what the downstream pathway could be of this. So knowing the downstream pathway of integrin alpha-5, they looked at FAC, AKT, and ERK. And you can see that as the concentration of AIB5P increased, the concentration of phospho-FAC, phospho-AKT, and phospho-ERK also increased, suggesting that the phosphorylation of these downstream messages were implicated in the signaling of this peptide. They found also a decrease in ALP and Alzheimer's red staining when they inhibit FAC activity, which was what they assumed was the downstream effector of the AIB5P molecular mechanism. So this confirms their hypothesis. So from this section, they could conclude that the AIB5P works by binding integrin alpha-5, which then leads to FAC phosphorylation and AKT and ERK phosphorylation, then upregulating genes associated with osteogenesis. They could completely conclude that they successfully developed a mechanism to learn functional oligopeptides from the IDRs of proteins with roles in bone formation, and that this approach can be applied to many biological processes for the development of further oligopeptide drugs. Thank you for listening.
Very good, Anna.
Thank you.
Good job, yes.
Very clear.
Thank you.
And also perfectly in time. So, okay, maybe, okay. I don't know if there are some questions. Let me check in the chat. Not yet. Okay, so maybe first of all, if I can ask Professor De Gasperis maybe to expand a little bit on the first part, just to clarify to the audience better than as we can do, how this process works.
Yes, since this work is based on unsupervised machine learning, probably it's better to understand what is supervised machine learning. In the example that Anna did with numbers, that where you have to recognize the pattern of a handwritten number and to associate with a symbol, that's exactly a supervised context, because you are imposing to a neural network the input and the output, and say, given this input, you should give me this symbol. And in iterating many times, the neural network will adjust its internal weights between neurons. So it's a very simplified model of how biological neurons work. It's definitely not the same, okay? It's a mathematical model. On the other hand, when it's unsupervised, then the supervision is kind of self-generated. In this case, it's based on probability. There was a picture where the neural network was showing the last layer of neurons were called softmax, right?
Yeah.
Softmax is a specific layer of neurons that has a specific mathematical property that the activation function of each neuron that goes between 0 to 1, the sum all over the neurons in the last layer should be 1, which means that each neuron activation will emulate a probability value. So basically having, I don't know, maybe 100 neurons on the softmax layer, you have a 100 discrete intervals of probability. And then the system will tell which one of those oligopeptide sequences are most probable to be functional given a specific biological pathway. So then there is another software on Cascade, which is the Monte Carlo simulator and the sequence generator that actually start from the output of the neural network to generate possible sequences of amino acids, taking into account what is the most probable that was selected by the neural network. Is that more clear or more confusing?
Yes, thank you.
Yeah. Yes, very nice explanation. Okay, so since I can't see any raised hand or questions, I can make some question to Hannah. I had some curiosity. First of all, you mentioned at the end that they said that could be future application of this method. And did they mention some other application, or do you read something about this? So the peptides generated by this method could be used for other stuff, other issues.
Yeah, I think a good example of this, a specific example, is cardiac diseases. So you could take a subset of proteins that have involved in cardiac activity, take the RDRs of those and generate oligopeptides that could help cardiac diseases. I don't know much about this topic, but this was mentioned. And also, I wrote down, for targeted drug delivery. So because the IDR protein oligopeptides, they go to, well, it was shown in this, they can bind to specific subcellular locations or specific cell types. You could use this, you could somehow conjugate this with a drug that can go to a specific cell type and be delivered directly there. I think that could be to other applications of this method.
Yeah, maybe cancer therapy or something like that could be very useful to target cancer cells, maybe.
Yeah, for the drug delivery.
Using this peptide as a vehicle, let's say, to target.
Yes.
Can I make a consideration? This is actually interesting how information has an impact on biology and also on a generation of new treatments.
Yes.
This is the first time actually that a software-generated peptide has a positive impact in a biological pathway. So this is a very milestone paper, I think. Students should study in detail and see how this research will go over the next few years.
For sure.
There will be many, many more applications on this kind of approach.
Yes, for sure, Professor. I completely agree, because bioinformatics and marriage between biology and informatics stuff, artificial intelligence is the future for sure. We are going this direction also by deep sequencing analysis, also in the RNA part, and yes, for sure, you are completely right. So we have, oh, sorry, Anna, do you want to comment on this?
I think this is Oriana using my laptop, so it says Hannaford, but it's not me.
Okay.
Self-supervised question.
Okay.
Yeah.
Okay, maybe I can read. So I think Oriana said that I'm curious how long this process takes to generate a set of peptides to investigate experimentally. Did it say in the paper any mention of timing?
They didn't actually, so I don't know. They said where they ordered the peptides from. It was something like China peptides, but I have no idea how long this kind of thing takes. I think it's quite a complicated process. So, and especially as they wanted well, they asked for 28 peptides, which is, you know, it's not just one. So I assume it was fairly time consuming and probably quite expensive process, but timings, I actually have no idea. I'd have to look into that myself.
Mm-hmm. I can add that since this is a deep learning context, usually these kind of networks have many intermediate layers, which means that if you try to do the learning on your own laptop, for example, it may take days. And so for doing this, you usually use a rented cloud computing platform, which has GPUs. They're used for video games, right? Or if you have a friend which is a gamer, you can ask him to lend you its workstation for an afternoon or something, and so you can reduce from days to hours to do a proper deep learning cycle.
Oh yeah, if I can add, also let's say more to how to prepare the process than to analyze. Maybe it takes more time. I mean, in the term of deep learning system. So how to training the process and maybe how to set the experiment before it takes more time.
Well, definitely the data set construction, data cleaning, data preparation is at least 50% of the effort of research.
Mm-hmm. So it's a very hard work, I think, the first part.
And in this case, you have to consider all of the subsequent in vivo, in vitro, and whatever other analysis in the laboratory. So
sure, for this for sure, you should set up the experiment before and then perform the experiment that replicates to have statistics. So yeah, it's not easy, but you know, research is not an easy task. So, but for sure, I can see a very gold future in this, I hope.
Maybe I can say at the current state of technology, you may think one PhD time life to do a new AI-based treatment based on peptides.
Mm-hmm. Okay, I can see Katie that raised her hand. So please, Katie, unmute yourself.
Hi, I wondered if you had any thoughts about doing combination therapies with this technique here. It seems if I understand correctly, they only went for one peptide in this study. But if you could say use a combination therapy to be very specific with the treatment plan, if there was a certain problem that needed targeting, it could be potentially quite interesting to then investigate using different peptides in combination together to produce whatever outcome it is that you're looking for.
So
do you have any comments?
Of the 28 peptides that they developed, choose more than one of these, you mean, in this example?
Yeah, for this example, or for other treatment plans, it could potentially be quite good to have a precise treatment plan for the patient.
I don't quite know why they only chose one. I guess it achieved the results that they wanted. But maybe in other experiments, if for a different area of biology other than bone, maybe the cardiac ones, if they need a specific outcome and one peptide does kind of half the job and the other one does half the job, then I think that would make sense. But they didn't really discuss this in the paper at all. They didn't mention why they didn't ever consider using 2 peptides for the rest of the experiments. So, but I think it would make sense to do that.
Yeah,
we'll have to see what other papers will come out.
As a first step, it's definitely useful to concentrate on one result only and one single. I would use a robotic keyword, which is actuator, right? So in this case, the total number of unknowns is only related to one single actuator. which is that specific peptide sequence. If you add another one, the number of unknowns explodes.
Yes.
So it may be difficult to compute. Okay.
Yes.
Also, one of the critical stuff could be when they will test this peptide for side effects. I don't know because they didn't mention this, but oh yeah, they did. Sorry.
Yeah, they found. Oh, you need to mute.
Oh yeah, sorry.
They found, yeah, they tested on other organs and they checked and they seemed normal compared to controls. And again, on immune cells, there were no differences from this. So they did check this and it was all fine. So yeah, I think as well, because proteins have such short half-lives in the body, they don't kind of stay around long enough to have these long-term effects. They don't get built up in the body because the body degrades the proteins. So this is another positive of using this technique, I guess.
For sure. Okay, again, Oriana said, is this paper first, a first for this method, like a proof of concept?
I think so.
Maybe, yeah, I think not for peptide-based paper, but for deep learning method, yes. I think it was the only one that to use this, sorry, the right word.
Unsupervised.
Unsupervised, sorry.
Yeah, also considering all the rest. I mean, considering everything is in the paper, integrated, like how they choose from NLP, natural language processing algorithms, from literature, the names of proteins in the first part of the paper, right? All related to bone diseases. That is more like a text mining kind of research. And then deep learning, and then in vivo, in vitro, as an integrated paper is the first one. Yes.
Yes. So it's very nice.
Yeah, so kind of in the conclusion, they were saying, Michaela, they were saying that, yeah, for other conditions, this technique works. They proved in bone that it works, not so much to use this peptide as a treatment for bone, but they didn't, this wasn't the main focus. The main focus was this concept works. You can take a condition and give the deep learning system the types of peptides that you want, and it will work and produce the peptide that has the desired effect.
So the method itself is the result of the paper. The
main
method.
one, yes. They said that their peptide works in bone formation, but their kind of overarching conclusion was this can be used for further diseases, etc.
Yes, I think the main news of this paper is the method, not more on the techniques used for the confirmation. It's more on the selection of the peptide. Okay, so please, Katie, again.
Do they have any intentions in future papers to take this peptide forward? If it was so effective in this paper, or is another group going to take up this as a potential treatment? Do we know?
I don't know. They didn't mention this, and I haven't seen anything about this. I don't know if Michaela knows, but just other than the suggestion to use this technique to develop more peptides for other diseases, there was no mention of using this peptide as a treatment for bone loss. It was suggested, but nothing solid. Michaela?
Well, they didn't mention, as Hannah said, so for sure, we don't know what is the future of this regarding this group, but for sure this group or other people will follow this, and because it's very promising, I think. So I'm hoping that it will go on with this, or other peptides. Should be nice also to test the other, or if they can find any other mechanism that could be used for maybe for other, or also bone diseases, you know, because it's not it's not only on osteoporosis or fracture. There are many other bone diseases, also maybe rare diseases. Why not? So for sure, it could be very useful also in the bone field, I think, to go on with this. Welcome, Katie. Thank you. So another, Oriana is very curious today. So she says, do you know the background of the author, please? As an interdisciplinary paper, I am curious about who contributed to the work. It's a nice question. I don't think we go through the CV of the authors. I don't know if Professor De Gasperi maybe.
Yeah, some of those are names from machine learning, but there are 16 people signing the paper. So it would be quite a big team. So it would be nice to do maybe in another meeting after we've been data mining their publication to know.
Sure,
exactly.
Maybe it should be very nice to have more on computer science because it goes through all the disciplines. It's quite, spread. It's going more and more spread. Yeah, so for now, we can't answer your question, Horiana, but it should be nice to go through and understand well. But for sure, it is in an interdisciplinary paper. So for sure, there are people that work on the biology part, people that work on artificial intelligence part. And I think it's the future to have teams that have many different skills and person, and you can see what is the results. So very great paper and very great advance. News.
Don't forget engineers, because you still have to keep up with the cloud computing architecture and facilities. So somebody like just a system administrator will be crucial to the success of this project.
Yes, for sure. Thanks, Professor. Okay, so since we have more minutes, maybe I can ask one thing. So I know, because I read the paper, that they buy their peptides from an industry, but do you know how artificial peptides are made, generally speaking? I mean, I don't know if you read something about this.
Yes, so they anchor the first amino acid onto a surface and then have washings, add the second amino acid. They can either do it in kind of an excess model, so they add a large amount of this amino acid in a solution, hopes that one amino acid attaches, and then have washings. Again, repeat the process until the peptide is created. I think they anchor it to a polymer, some sort of polymer they anchor it to, and then extend it this way. Which is why I think when Oriana asked her question before, I misinterpreted it, thinking that she was asking how the proteins were physically made. This is why I was explaining it's a time-consuming process, and I think quite expensive, which is one of the downfalls of the paper that they discussed. So yes, I think it's like this, but again, it's something that I don't know much about.
Yeah, sure, because also they didn't mention how they made it, because they said that in the meter that they buy this peptide from an industry. For sure, I think could be the method. And also related to this, I would like to ask you why they didn't use a natural occurring peptides instead of an artificial one, because peptides could be found in the body, in the cells. But they use artificial one.
I think that the benefits of this, obviously it was a proof of concept that this technique works. Also, using artificial peptides, you can be sure that it is purified. If you take it from a naturally occurring in the body, you can't be 100% sure it's purified. There's all of the ethical issues surrounding that as well. Generating it yourself, you can modify it. So they discussed modifying the peptide to have it last longer in the body, have a longer half-life. You can do the targeted drug delivery, you create the peptide whilst attaching the drug vehicle. So many reasons. And maybe to also generate something that works better than something that naturally occurs. You could create a peptide which actually functions better than anything that naturally occurs in terms of peptides. So I think these kind of reasons why.
If you can add, so the result of the paper, the AI P5, whatever, is an artificial peptide, which was born in a computer memory. So you then need to synthesize it to make experiments.
Sure. You're right, Professor. And also, I think you could also, if you purify, maybe you have, I don't know, modification of the peptide that occur in the body for many reasons. So maybe you should, I don't know, again, made it artificially after the purification. What I'm thinking was the, I know, post, I don't say, transcription, yes, modification that occur in the body, in the cells.
Yes, this too is a good reason.
Okay, so I don't see any other question and any other raised hand here. I don't know if you, in the audience, we are a lot, so maybe some of you can have also naive questioner. Welcome, because we are all naive basically on this field, so we will try to discuss on it. Maybe we don't know, but we can try to hypothesize or speculate a little bit on this topic, because it's nice to discuss together. Yeah, I'm thinking about something else, just to, yeah, one question could be, did they describe the lowest effective dose of this peptide? There is a dose, a lower dose to be functional.
There could be, but again, they didn't quite describe this. As this paper, as Oriana said, was kind of more of a proof of concept, they didn't describe the lowest effective dose of this, which could have been quite interesting to know. If it was particularly low, it would be quite nice. But obviously you could see from the ALP staining and the Alzheimer red staining that the higher doses clearly had higher effects. So it would kind of be a case of what counts as a significant enough effect. Where would the threshold be to be like, it will probably increase bone formation at all doses, but by a tiny amount. So it's the case of deciding what is enough to be classed as the lowest significant dose. So yes, they didn't do this, but you can see it does increase over the concentrations which they tested.
Yes, for sure, it's a dose-dependent effect. It's not just the peptide is there and works for everything. So for sure. Okay, so we have 10 minutes, so don't be shy, please, people. Otherwise, I can wait just 1 minute more. And meanwhile, maybe I can thanks Hannah for the hard work and Professor De Gasperis for all the help and the clarification that he gave us. Also, thanks for this inspirating paper that could be nice to have any more ideas also on our work. Why not? Yeah, so I can't see any further questions. So I can thanks Johanna, Professor De Gasperis, and everyone here for being here and listen to our journal club. And yeah, we can see you next Thursday. Thank you all.
Thank you.
Thank you. It's been very interesting.
Thank you.